Hello, hello. Welcome to the online lecture again. Today we will talk about the molecular basis of evolution. So the first main point you need to know is that evolution affects the population, not individuals. So we'll talk more about this on Wednesday's lecture. We'll talk about microevolution today, which is the first of the 3 evolution lecture, which is not very common for most of the classes. The reason I do the microevolution first, because we just finished genetics, and microevolution has a lot to do with genetics. So that's why I use microevolution as the first evolution lecture, so that you will have a easier transition. So first of all, you have to understand that evolution affects a population, not individual. This is the first main point. And at that time, even though Darwin observed the evolution occur in the nature, he could not explain how the trait of animal change over time. The reason is that he does not know anything about genetics. Even though Darwin and Mendel they are living about the same time, Darwin published his origin of species like approximately 6 years earlier than Mendel's published his genetic work on the pea plants. So by the time Mendel published his work, Darwin is already famous because of his natural selection evolution idea. So at that time, 6 years after he published his Darwin's idea, Mendel sent his paper to Darwin. But for some reason, Darwin didn't read Mendel's finding, didn't read Mendel's paper. Mainly because after he got famous, a lot of people sent their paper to him. Yeah, a lot of people, once you get famous, a lot of people will send the paper, their scientific finding to Darwin to ask for his opinion or something like that. So Mendel is just one of the many people who send their paper to him. And of course, Mendel, and most importantly, Mendel's paper has a lot to do with math. And Darwin, at that time, think that math is not suitable, it's not useful in biology because, well, his evolution idea is based on observation in nature. So when he looked at Mendel's paper, even if he did look at Mendel's paper, he will probably throw it away because it's all about math and probability. So I would say this is the very sad story for science. But nowadays we are able to we put these two together, especially we put Mendel's work first, because we want you to know the underlying reason for what happened to the evolution. Basically, Mendel's work is the basis of evolution. Genetics is the basis of evolution. So that's why we teach you genetics first, and then after you understand genetics, then when we talk about evolution, you'll have an easier time to understand. And not only that, I deliberately talk about microevolution first as a transition for you from genetics into evolution, because microevolution has a lot to do with genetics. So Darwin cannot explain what he observed. How come the trait will change? The reason is that he doesn't know anything about genetics. Of course, nowadays we know that genes interact with the environment, or environment affects the trait. Evolution, the third point is the most important for this slide here. For this slide here, he said that it's really about genetic changes. The gene and trait, they are linked. Of course, the environment can exert an effect of the expression of certain trait. We'll talk about how environment can affect the expression of trait. Now, this is the definition. Microevolution has to do with the evolution changes within population. Again, it is population, not individual. Microevolution in pepper moth. We use pepper moth because in the next lecture we'll talk about an example of the pepper moth. What happened when they are during industrial revolution? So industrial revolution causes let's talk about this and then I'll ask you a question so that you can pay attention to the Wednesday lecture. What will happen there? Population genetics is a field biology that studies the diversity of population at the level of gene. Again, it is a population, and it is diversity, and it is a gene. It has something to do with gene. So that's why it is population genetics. And we briefly talk about the definition of population in our first class, the first lesson. So population geneticists, scientists who study population genetics, they're interested to know how the genetic diversity in population change over generation from generation. We will see several examples in today's lecture about the changes in the population, and also there are different types for that too. So microevolution is to measure the diversity of the population in terms of allele, genotype frequency, most important, genotype frequency. Allele frequency and genotype frequency. These are the two frequencies that microevolution study. So I'll show you a picture that summarizes all this. We will tell you what do I mean by allele frequency and also genotype frequency. So we use pepper moth as an example. So pepper moth is just like the pea plant that we talked about previously. They have two alleles for a gene, for a single trait. The single trait is the color of the body color. The big D is the dark color, small d is the light color, and big D is dominant. In other words, the allele will be the genotype, the possible genotype, the big D, homozygous dominant, big D, small d, heterozygous dominant, and small d, homozygous recessive. So the dominant one will produce a dark morph, the recessive one will produce a light color morph. This picture, I like this picture because it summarizes basically the previous two slides. So you can see that, okay, homozygous dominant, heterozygous dominant will give you the dark morph. Homozygous recessive will give you the light color morph. We call it pepper morph because it looks like a white morph, and then we spray pepper on it. Then you see all this black dot on the morph. They all locate the allele locate on the same locus. Gene pool, this is another definition you need to know. It's the sum total of all alleles of all genes in a population. So basically, it's everything is included. Sample study. Sample study. Let's say we have 25 pepper moth from the population. Of course, some of them they're dark, some of them they're light. For these 25 pepper moths, we have 50 alleles. One, because 2 alleles for each moth. Then we have 25 moths. So 2 times 25, we have 50 alleles. In that, we found that, okay, this is like given. We found that 10 alleles, they are big D, and 40 alleles, they are small d. So how do we find the frequency? Yes, it has something to do with math and probability. So how do we find out the frequency of the big D allele and small d allele? Since we have 10 alleles that are big D, that means it is 10 over 50, the total. Then it is 0.2% or 0.2 or 20%. 0.2 or 20% chance to have the big D allele. And 40 over 50, we will get 0.8 or 80% chance. We will get the we have 80% chance to get this small d allele or recessive allele. So this 20%, 80% tells you the probability or the proportion here of the allele in the population. And this one is important. The frequency, when they add up, they should always equal to 1. They should always equal to 1. So we can use this expression P plus Q equal to 1. P is the representative frequency of the big D. Q represents the frequency of small d. So the frequency of the dominant allele plus the frequency of the recessive allele, it should be equal to 1 all the time. Last point is actually important for you to understand why we do all this math. What's the purpose? Because we want to see if there's any changes in the population of a moth in this case, or so if there's any changes in the allele frequency, that means if P is no longer 0.2, Q is no longer 0.8, then we can assume that there is evolution occurring. And here, if the allele frequency, that means P is always 0.2, Q is always 0.8 for, let's say, 3 generations, then that means there's no evolution occurring in that population. So by looking at the allele frequency, P or Q, then we can tell whether or not that population has evolution occurring or not. So Hardy-Weinberg equilibrium or Hardy-Weinberg equation. So this basically tells you what I just told you. If the allele frequency do not change, that means the population, the allele frequency in the population achieve a Hardy-Weinberg equilibrium. That means the population is stable or non-evolving. That means the population is not evolving. We call it genetic equilibrium. So remember, when the allele frequency of a population do not change for a long time, then that means the population is not evolving. We call it genetic equilibrium or Hardy-Weinberg equilibrium. So what is Hardy-Weinberg? So there's something called Hardy-Weinberg equation, which is this one. This is what we call the Hardy-Weinberg equation. So you can see that this one is the dominant allele, Q is the recessive allele, and PQ will be the heterozygous dominant. So in this case, the Hardy-Weinberg equation is to calculate the genotype. Okay, it is not the allele frequency; it is the genotype and allele frequency. Well, they said both. So here you need to know the allele frequency first. That's what we did previously. The allele frequency here is 0.2. So if it is p squared, that means it is 0.2 times 0.2. Then that is the genotype for the homozygous dominant. For the heterozygous dominant, big D, small d here, it is 2 multiplied by 0.2, p equal to 0.2, right? The allele frequency for the dominant allele. Q is the allele frequency for the recessive allele. It is 0.8. That means 2 times 0.2 times 0.8. Then it will be the genotype frequency for heterozygous dominant. Last one, Q2, which is the 0.8 times 0.8, which will be the homozygous recessive genotype of frequency. So this picture summarizes what I just told you. So we have dark morph. They can be homozygous dominant or they can be heterozygous dominant. No matter what, they have the dominant allele, big D. And for the heterozygous dominant, they also have the recessive allele, small d. For the light color morph, they are all homozygous recessive, so they all have this small d allele. And then remember the allele frequency. Allele frequency is this one, p plus q equal to 1. Allele frequency, p is equal to the frequency of the dominant allele. Q is the frequency of the recessive allele. So remember, how do we do it? It is, for example, if we have 10 of the big D allele, and the total is 50 allele, then it is 10 over 50. And if the recessive allele, we have 40 small d, and the total allele we have is 50 total, that means both dominant and recessive together with 50, then the frequency for Q or small d, it will be 40 divided by 50. That's why we got 0.2 and 0.8. 0.2 for P, 0.8 for Q. And the sum is always 1. For the Hardy-Weinberg equation, it is this one, P squared plus 2PQ plus Q squared equal to 1. Again, the end result should also be equal to 1. So P and Q, they're the same for both, but the frequency of the homozygous dominant genotype, the frequency for the homozygous dominant genotype is p squared. So as I said before, it is 0.2 multiplied by 0.2, which will be equal to 0.04. And then the frequency for heterozygous dominant genotype will be 2pq. So 2 times 0.2 times 0.8. Then we'll have 0.32. And then the frequency of the homozygous recessive, which is q squared. In this case, the slide has a typo error. Theoretically, it should be 0.8 times 0.8, but the slide put it as a 0.08. So that's why I cross it and put 0.8 here. So q squared is equal to 0.8 times 0.8, which is equal to 0.64. You add these three together, you will still get 1. So this is how we find out the allele frequency and using this p plus q equal to 1, and also the genotype frequency. Genotype frequency, we use Hardy-Weinberg equation. Allele frequency, we just use this p plus q equal to 1. So remember, they are allele frequency and genotype frequency. We use different equations to find out, to calculate. So for Hardy-Weinberg principle, the allele frequency, they will be at the equilibrium if and only if all these 5 conditions are satisfied. So you must have all 5 satisfied, then you so-called achieve the Hardy-Weinberg equilibrium or genetic equilibrium. What does it mean, genetic equilibrium or Hardy-Weinberg equilibrium? It means that the allele frequency does not change. That means that the trait or the genotypic frequency also does not change. No mutation occur, no genetic drift, no gene flow, no random mating, no selection. I will explain all this. What does it mean, mutation, genetic drift, gene flow, random mating, or selection, natural selection. In the next few slides tonight, I'll explain each one. So don't worry about this. As long as there are no, and then it is a random mating, and then no selection, then it will be no natural selection. Then the population will be in an equilibrium. That means no evolution occur. No evolution. It said that it is very, very difficult to have 5 conditions all met. That's why it is rarely occur. In other words, in a population, evolution almost always occur, either in a large scale or small scale, either in a faster pace or a slower pace. But evolution always occur because it is almost impossible to have all 5 conditions satisfied. So this Hardy-Weinberg principle, why is it so important for us to talk about? Because it tells you that microevolution can be detected by looking at any changes in the genotype frequency and the allele frequency for population. So we'll talk about the 5 conditions now. So if the 5 conditions are met, then the population will achieve genetic equilibrium. If one of the 5 or all 5 or if any of the 5 conditions they are changed, that means it is the opposite. Or any of the conditions are changed, then evolution occur. So let's talk about each one by one. Mutation. Mutation, well, of course it can cause the allele frequency in a population to change. And the second point is very important. Mutation is the only source of a new variation in a population. Yes, mutation is basically can literally I don't want to use the word create, but it can produce. Mutation can produce a new genetic variation, that means a new trait in a population. It said that without mutation there will be no new genetic diversity. Yes, mutation is the one, as I told you, is the one that produces a new trait. Mutation, a random event. Yes, mutation occur in random. I remember in X-Men there's an episode that Magneto is trying to create a new mutant by, I don't know, by some kind of high voltage, whatever. So he's trying to create new mutation, but it is not that easy because the mutation is a random event. It says that some are beneficial, some are harmful, but I would add something to it. Some are beneficial, and then you put a comma in here. Then most. And most are neutral. Yeah, for mutation, of course, some can be beneficial, some can be harmful, but most of the naturally occurring random mutation, they are neutral. That means they are not harmful, they are not beneficial, they are just doesn't do anything. Well, if it is, the truth is that our gene, our body, they have a different mechanism to correct. Let's say if the mutation occurs and it is harmful, or if not harmful, if mutation occur, then most of the time our body have different mechanism to correct the changes in the DNA sequences. If any mutation occurs, if there's any mistake occur in the replication or transcription, translation, then some correcting mechanism we would have some basic mechanism to correct the mistakes, including mutation. Not including mutation. It's only for replication, transcription, and translation. Most of them are neutral because the mutation is random, so it may change only one amino acid sequence, then that sequence may or may not have any effect to it. Most of the time it may not have any effect because it is random. Then in this case, you will not have special power. First, after mutation, we'll talk about genetic drift. Genetic drift is a change in the allele frequency due to the random meeting of gametes in fertilization. When the random the change in allele frequency is due to random meeting of gametes in fertilization, that means the mating is randomized. Random selection and a sum of gametes in population causes allele frequency shift in each generation. It is similar to a mutation that it requires a randomization of the gamete and randomization of the selection and of the gametes. That means when you look at population, when you look at, let's say, the pepper moth in our previous example, if you look at the pepper moth, the allele frequency and the genotype frequency, let's say if you look at the population of pepper moth early in this, let's say in January, maybe you have the 0.2 and 0.8 ratio. But when you look at the pepper moth, let's say in summer, in August, then the 0.2 and 0.8 allele frequency may change. Maybe it will change to 0.3 and 0.7. And then when it is around, let's say, November or December, we look at the frequency again. Maybe it will become 0.25 and 0.75. That's what it means. Populations are always undergoing genetic drift. So it's just a matter of how much. It's not a matter of yes or no. So a population will always undergo genetic drift, but it may not be a significant change. It said that the change will be bigger if the population is very small. So you imagine that population is very small, then any small changes in the genotypic frequency, then you will have a very large effect, any big effect due to genetic drift. On the other hand, if the population is very large, that means you have a lot of moth, then when the genetic drift occur, then most likely the rest of the rest of the population can stabilize the changes in the genotype frequency. The reason is that the rest of the population, they may not experience the change in the allele frequency, or they may not experience the change of the genotype frequency. So when the genetic drift occurs, it may only affect part of the population. If the population is very large, it may only affect a small part of the population. But the rest of the population, they are not being affected. So they can mate with the part that is being affected, then in order to stabilize the allele frequency and also the genotype frequency. So if the population is small, then removal of gamete. What does it mean, removal of gamete? Removal of gamete is like killing part of the population. If part of the population is being killed, that means we call it removal of gamete of the smaller population. Then, of course, it will have a, because the small population is small, if you kill some of the member of a small population, of course, in terms of percentage-wise, you will have a greater effect to change the allele frequency and genotype frequency of that small population. This is one of the examples. For example, if, as you can see, the green frog and let's say the brown frog, the green frog, they are living on the lotus leaf. Let's say if there's a disaster killing a lot of lotus, so there's not a lot of lotus leaves in the pond, so that the green frog, they are forced to live on the land, on the ground. For this brown frog, they live on the ground. Originally, we have a lot of these frog, green frog, but natural disaster kills the frog. Let's say natural disaster kills the lotus leaf. So all the frogs, they have to move on the ground. What happens is that because the ground has this natural color here, so they may live better. Oh, sorry. This is a different analogy. So it said that the naturally killed the green frog, but the brown frog did not. The brown frog population is still stable. Then what happened is that only when you have a decrease in the population of the green frog, then you will be relatively increasing the population of the brown frog. So as you can see, we have one brown frog, and then 1, 2, 3, 4, 5, 6, 7, 8, 9 of them. So you have 10% of the population is brown, and then 90% of the population that is green. But right now you have one brown frog and 4 green frog. That means you have 80% of the population, they are green, and 20% of the population, they are brown. So by killing, or because of natural disaster, killing some of the green frog, then you can see there's a change in the genetic equilibrium. There's a change in the allele frequency, a change in the genotype frequency. So for under genetic drift, we have two different types. One is called bottleneck effect, one is called founder effect. Bottleneck effect is similar to what we just told you, the green frog and brown frog. So originally we have 2. Here it said that we have 4 different populations of the frog originally. But the bottleneck means that there's a natural disaster. When natural disaster occurs, only a few survivors can survive. Only a few of them can survive after the natural disaster. Then we take a look at the survivor of the natural disaster, and then we find that there's a change in the population of the genotype frequency and allele frequency here. You can see that originally we have 4 different types of population. Now we only have 3. You can assume that one type of the population, it goes to extinction. Extinction meaning that all population of a certain species are dead. And then the percentage is also changed for the surviving population. So this is what we call bottleneck. Founder effect, an example of founder effect is like the Amish people in the United States. Amish people, you know that they don't do modern technology. They are very they isolate themselves, and they only marry to other their own community. So their original gene pool is very large and diverse because they come from the old world, the major population. But then after they move to the US, they try to isolate themselves. Then they live by themselves. In this case, they form a very small population among themselves, and which is not a very good idea, because genetic drift can occur really. You can see the effect of genetic drift in such a small population easily. Then gene flow. Gene flow is the movement of a allele between population. So in summary, basically it's like migration or immigration and emigration. For an example, I give my students that it's me. I'm the obvious example. I was born and raised in Hong Kong, and then I moved to the United States. So I carry the gene from Hong Kong to the United States. This is what we call the gene flow. And the gene that the so-called Hong Kong gene that I carry, it will be mixed with the American gene eventually. Well, I mean, of course, if I marry American women, then it will mix with the American gene. But if I marry a Chinese woman, then if I marry a Chinese woman, then my gene will still remain to the Chinese. So it really depends on the population. Yeah. In this case, if my children they continue to marry to Chinese, then you may consider it as a founder effect, as another founder effect. Even though I bring in the Hong Kong gene from Hong Kong, but I stay with my own group of people, and my children also marry with their own group of people, then you may consider that as an example of a founder effect as well. But I can tell you that it's not the case. The ABC, American-born Chinese, they like to marry with other Americans instead of staying with with other Chinese. So if my children in the future, if they marry other American, then the gene flow mixes the genetic diversity, which is a good thing. Okay. In other words, gene flow, that means bringing in different foreign gene. It keeps the gene pool of 2 or more populations similar, because they mix, and then eventually the 2 populations will reach another equilibrium of the differences.